be by Joel Sharbrough, and he's going to be telling us about the effects of mutation drift equilibrium.
Hi everyone, I'm Joel. Thanks for coming. So today I'm going to talk a little bit about my PhD work with Maureen Neiman at the University of Iowa, working on the evolutionary forces that contribute to, or perhaps protect from, mitochondrial mutation accumulation, using this wonderful snail, Potamopyrgus antipodarum. So, I probably don't need to convince anybody in here about the importance of mitochondria, but I would like to say that the forces that contribute to mitochondrial evolution are not straightforward. Firstly, as we heard from David, the production of reactive oxygen species is going to cause high mitochondrial mutation rates, especially in animals. So you have a high input of mitochondrial mutations. Secondly, and we've heard a lot about this today and throughout the conference, is the interaction between two different genomes to produce a critical component of eukaryotic health, that is the production of ATP, means that the mitochondrial genome and the nuclear genome need to match up and function properly together in order to, for these organisms to survive. Now, thirdly, and I'd argue critically, sex plays a major role in this. Now, sex is only occurring in the nuclear genome. However, if we remove sex from the nuclear genome, there can be major effects on mitochondrial evolution. So, just as a cartoon introduction to this, and very simply, let's assume that we have this female here with a blue nuclear background. These X chromosomes just mark different nuclear chromosomes. And then it's got this circular purple mitochondrial genome, and then some mutations present on her genome. So she'll mate with some male, and he'll have a different mitochondrial genome with his own mutations and his own nuclear genomic background. The offspring, this daughter, will have them generally, there's obviously exceptions as we have heard, generally have the mother's mitochondrial genome, along with an assortment of mutations from the mother and father, but not inevitably inheriting those mutations from them. So sex offers a way to redistribute mutations on the nuclear background and place mitochondrial genomes on different backgrounds. And over the course of generations, this can result in mitochondria moving across different nuclear genomic backgrounds, but we don't necessarily expect that mutations will accumulate as rapidly given it's moving to these different backgrounds. On the other hand, in asexuals, the mitochondrial genome is co-transmitted with the nuclear genome. So, every mutation that the parent had gets passed on to her daughter, with the exception of back mutations. So you get this effective coupling between the nuclear and mitochondrial genomes that's going to have some fairly complicated evolutionary consequences. I'm not going to talk about all of them today, but I do want to mention some of them. So one potential consequence out of this is that epistatic variation becomes genetic variation when these two genomes are linked and could potentially be acted upon by selection. Secondly, you can have things like more powerful Muller's ratchet type effects where you get mutation accumulation due to the elimination of the least loaded mitochondrial mutation class. And thirdly, and this is what I'm going to focus on today, you could get Hill-Robertson type effects where selective interference between loci can decrease the efficacy of selection. So, in other words, mutations can no longer be selected independently from their genetic background. So in the absence of sex, what we expect is that mitochondrial genomes should accumulate mutations because of this co-transmission, and the nuclear and mitochondrial genomes are linked effectively, and so we should see elevated selective interference. So, to test this, we can use this really great model system, Potamopyrgus antipodarum. It's a tiny little freshwater snail, very cute, and they've got even better genomes. So what's really great about this species is that they're in their native range of New Zealand, they are polymorphic for sexual reproduction and asexual reproduction. And importantly, this polymorphism occurs within lakes. So you have sexual and asexual lineages coexisting and competing in lakes. as far as we can tell, they're phenotypically indistinguishable, such that they're competing for the same resources and doing the same things in these lakes. So it makes it a really nice comparison of mitochondrial evolution in the presence and absence of sex. Okay, so we have gone down this road a little bit, and so just to orient you to the mitochondrial genome of Potamopyrgus aspidarum. So it's a fairly standard animal mitochondrial genome with 13 protein coding genes. It's got all the tRNAs and rRNAs as well. I'm going to focus on just the protein coding genes. It's about 15,000 base pairs. The only weird item about this in terms of an animal mitochondrial genome is that it lacks a D loop. So we don't really know how transcription works in this species. It does get transcribed, we know that, and, but we don't know how it's regulated. But we've sequenced 31 whole mitochondrial genomes from Aspidarum and an additional whole mitochondrial genome sequence from a closely related sexual outgroup, Potamopyrgus aspidarum. So if we look at the phylogenetic distribution of these genomes, I want to point out a really critical part about this system is that we see multiple independent transitions from sexual ancestor to asexual descendant. And so we have these independent, not independent, but separate transitions to asexuality, which allow for comparisons across these different transitions to see if there's any common features of losing sex on mitochondrial evolution. I'll return to this phylogeny in a minute to point out some issues, but first I'd like to point out some of the progress we've made so far. So in 2010, Maureen published this paper demonstrating accelerated mutation accumulation in asexual lineages in their mitochondrial genomes. And so what she showed was that a couple trees right here, so the same topology of these trees, but the branch lengths are synonymous substitution rate on the left and non-synonymous substitution on the right. Two critical inferences from this is that the branch lengths in the synonymous tree are significantly longer than the branch lengths in the non-synonymous tree, meaning that non-synonymous changes are generally selected against. And secondly, that the asexual branches, and especially in this non-synonymous tree, I think we're running out of juice on this laser pointer, but especially in the non-synonymous tree, the branch lengths are significantly longer in the asexual branches than in the sexual branches. So non-synonymous, putatively harmful mutations accumulating in these in the mitochondrial genomes of these asexual lineages. More recently, we're putting up this paper where we found that radical amino acid changes persist longer in the absence of sex. This paper is in review right now, but it's on bioRxiv if you want to check it out. The major upshot of this is if we consider conservative changes in this sort of light blue color and radical changes in this dark blue color in only sexuals, in terms of the number of unique mutations per mitochondrial genome, what we see is that sexuals can effectively distinguish between different classes of mutations. So really severe mutations are being selected out of populations. They're getting kicked out of populations more rapidly than minor, relatively minor mutations. If we look at asexuals, that's not the case. They cannot distinguish between conservative and radical mutations, indicating that purifying selection is not acting as effectively in asexual lineages. Okay, so that's all from a genome-wide perspective. Today I'm going to take you on sort of a gene-by-gene trip through this. And in particular, we have to address some of the problems associated with this system. So I love these snails, but there are still some problems. And one of the major problems is that the transitions to asexuality are not phylogenetically independent. So we have to do sort of a clade-specific approach. We don't have enough transitions to asexuality to do some classic Felsenstein-esque phylogenetic independent comparisons. But I'm going to try and convince you that we've made an attempt to address that. Secondly, is that genetic variation in this species, in the mitochondrial genomes of this species, is structured. So the only nodes on this tree that I'm showing are nodes with greater than 80 bootstrap support on a maximum likelihood tree. So we're pretty confident in these nodes, And what that means is that there's a lot of genetic variation that is private to certain clades in the species. So mutations that are present in this group are not accessible to this group. This is compounded because asexuals aren't sharing alleles anyways. So we've got to deal with this structure somehow. So we've come up with a couple of approaches, and we're working on a couple more as well. One of them is using PAML, phylogenetic analysis by maximum likelihood, to estimate branch-specific rates of non-synonymous mutation accumulation, DNDS. Also, I've developed a program that infers population genetic statistics and does clade-specific bootstrapping. The name is obviously up for reconsideration, so if you have any suggestions, I'd happily take them. For the rest of the talk, I'm just going to refer to them as PAML and SBC. Okay, so let's look a little bit at these genes. We've got a couple different measures of natural selection between species, within species, and then using a polymorphism and divergence measure. And this is using the SBC method. What we see in all of these genes, in terms of DNDS, is extremely low values of DNDS, indicative of strong purifying selection acting across the genome. If we look at a more recent timescale at polymorphic level, we see theta N, theta S values, which have the same interpretation as DNDS, although you have to account for demography potentially affecting these. But we see very similar patterns with all 13 protein coding genes exhibiting low levels of polymorphism in their non-synonymous sites relative to their synonymous sites. So again, indicative of purifying selection. If we look at a polymorphism to divergence timescale, so using McDonald-Kreitman tests, what we see is that there are significant values of purifying selection for 7 of the 13 protein coding genes, with purifying selection being the case for all of those. If we combine genes into their complexes just by concatenation, we see very strong signals of purifying selection in DNDS, theta N, theta S, and McDonald-Kreitman tests. So it's clear that selection is operating here, but is mutation drift affecting this as well? One way to get at this is by comparing the synonymous substitution rate to the non-synonymous substitution rate. If selection is operating and drift is not important, then we shouldn't see a correlation between these. However, if we look across these genes at branch-specific estimates of DN and DS, what we see is DS is highly correlated and significantly correlated with DN, indicating that mutations do contribute to mutation rate does contribute to non-synonymous variation, but it doesn't explain the whole story. So, and I have some additional data that can point towards that. If we look at the number of polymorphisms in in each gene relative to the number of substitutions, if it were purely mutation rate driving non-synonymous variation, we'd see a relationship, something like this, highly correlated, and that's what we see in the synonymous changes. But when we look at the non-synonymous changes, we see a significant and positive correlation, but at a significantly lower level than the synonymous changes. So mutation rate is playing a role, selection is playing a role here. Okay, so if we look across the complexes, we see a very similar thing, is that selection is driving molecular evolution across complexes. So perhaps the unit of evolution, and on the top I'm showing divergence levels, on the bottom I'm showing polymorphic levels. Selection is mediated according to complex. And then one final slide is that, it appears that genes that are under more stringent selection accumulate mutations at a lower level than genes that are under less stringent selection in asexuals. So we think that the selective constraint determines the extent of mutation accumulation in asexuals. I'd just like to thank some collaborators, especially Maureen, and I'll take any questions after. Thank you.